Hello, my dear students. Welcome you all to Shri Jayendra Rank Zone classes. I am a mentor and your friend for the subject biology, and I will be taking care the animal science, that is the zoology section. My dear students, in the last videos, we have started the introduction of animal tissues. We have completed the classification that how animal tissues are further classified into different categories, and now we have been begin previously for the epithelial tissues. Now we are going to study about some specialized functions or some specialized modifications shown by epithelial cells. These structures which are actually executing a specialized function, they are very important as their location been a very favorite question of Neet examiners to put in the question paper. So coming to specialized function structures shown by epithelial cells, microvilli. Microvilli, they are minute processes, they are minute protoplasmic processes, okay, which are non-motile. They are non-motile, they are minute protoplasmic processes, and they are non-contractile. You have to remember these three terms: minute protoplasmic process, non-contractile, and non-motile, right? They help to increase the surface area, more than 20 times. Okay, they help to increase the surface area by more than 20 times. They help in absorption, secretion, and excretion of food. They help in absorption, secretion, and secretion of food. Okay, present in the wall of intestine. They are mainly present in the wall of the intestine. They are present in the wall of intestine. Gallbladder, proximal convoluted tubule, PCT, also known as PCT, proximal convoluted tubule. So microvilli, they are minute protoplasmic process. They are non-motile, they are non-contractile. They help in absorption, secretion, and excretion of the substances. They help to increase the surface area by more than 20 times, and they are present in intestine, gallbladder, and PCT, proximal convoluted tubule. So is that clear? Now, coming to next, cilia or kinocilia. Cilia or kinocilia, they are long cylindrical processes. They are long cylindrical processes, protoplasmic process. Diameter is same from base to apex. Okay, diameter is same from base to apex. From base to apex. Okay, they are motile, they are contractile. Okay, just opposite to microvilli. The movement of cilia is always in uniform direction. The ciliary movement always happens in uniform direction, okay, in a single direction. So cilia or kinocilia, they are long cylindrical protoplasmic processes. The diameter is same from base to apex. They are motile, they are contractile, and the ciliary movement, it happens in a uniform direction, in only one direction. Cilia or kinocilia, they originated from the basal granules. They originated from basal granules or kinetosome. Okay. So the part which give rise to these ciliary processes is basal granule or kinetosome. Understood? They help in the movement and locomotion. Okay. They help in the movement and locomotion already mentioned. It moves particles or mucus in specific direction. It move, helps to move particles or mucus in specific direction. Understood? Over the epithelium. Movement is in uniform direction, right? It is in specific direction means uniform direction. The diameter of cilia is same from base to apex. Okay? In internal structure of cilia, 9 plus 2 arrangement of fibrils, microfibrils is seen, arrangement of microtubules is seen, is present. They help in conduction. Okay. So the kinocelia or the cilia, they basically have originated from basal granule or kinetosome. The diameter of cilia from base to apex is uniform. They are motile, they are contractile. Okay, movement of cilia, it always happens in a uniform direction. It helps to move mucus in a specific direction. Now they are present in the inner surface of hollow organs like bronchioles and fallopian tube. Inner surface of hollow organs. Okay, it is present in the inner surface of hollow organs like fallopian tube, bronchioles. Okay, then they are present in trachea, uterus, ependymal epithelium. Ependymal epithelium is the inner lining of the brain cavities, means the ventricles. Okay, so the inner lining of the ventricles means the brain cavity is ependymal epithelium and central canal of spinal cord. And central canal of spinal cord. Function of cilia is to conduct substances. Function of cilia here is conduct substances in CSF. Okay, in cerebrospinal fluid, the function of the cilia which is present in ependymal epithelium, it helps in the cerebrospinal fluid conduction. Conduction of cerebrospinal. Okay, so these are microvilli, cilia or canocelia. Now coming to next, stereocilia. Stereocilia you can see in this picture, these are cilia and these are microvilli. So microvilli you can see the finger-like projections. Cilia, they are hair-like projections. Okay, from basal granules they are arising. Microvilli, they don't arising from basal granules. They are, the overgrowth of the whole epithelium you can see. And stereocilia. Stereocilia, they are long cytoplasmic process which are conical in shape. Okay, long cytoplasmic process which are conical in shape. Conical shape. Okay, they are non-motile and non-contractile. So stereocilia, they are non-motile, they are non-contractile, just like microvilli. Basal granule is absent in case of stereocilia. Basal granule is absent in case of stereocilia. Plasma membrane is thick and rigid. Plasma membrane is present and it is thick and rigid. Base of the stereocilia is broad and apical part is narrow. Base of the stereocilia is broad and the apical part is narrow. So they are conical in shape. They increase surface area. Okay. Example, epididymis and vasa difference. Okay. So epididymis and vasa difference, these are the only two places where stereocilia is seen. Stereocilia is a false cilia. It is a false cilia. Okay, as they are non-motile, they are non-contractile, they are long cytoplasmic processes. They just help to increase the surface area. Means stereocilia looks like cilia, but they function like microvilli. Okay, so they are long cytoplasmic processes which are non-motile, non-contractile. Basal granule is absent in them. Plasma membrane is rigid and thick. Understood? And base of the stereocilia is broad and apical part is narrow. So they are conical in shape. They increase surface area. They increase surface area means they are present in epididymis and vasa differentia. Now coming to next, yeah, this is the picture which help you to understand the sorry, so sorry. Sorry. This is the picture which help you to understand the microvilli, cilia, cell junctions, and basement membrane. So you can see the microvilli, the finger-like projections. And you can differentiate microvilli from cilia. Cilia, they are very fine hair-like projections with the basal granule. Okay, but microvilli, they don't have any basal granule. Okay, you can see here these are microvilli, finger-like projections. Understood? Then you can see these are tight junctions, zonula occludens. Okay, then we are having intercellular spaces. These are intercellular spaces. These are intercellular spaces. Right? Then we have gap junctions. These are gap junctions. Okay, though gap junctions. Okay, these are gap junctions. Sorry, these are the gap junctions in which the cytoplasmic, exchange of adjacent cells takes place. They these radiating fibers are of desmosomes. Okay, desmosomes. Okay, made up of these microtubules or microfilaments are made up of keratin protein, right, and adherent protein, right. Then we are having intermediate filament. These are intermediate filaments and extracellular matrix. Okay, so everything is been shown here to basically make you understand that how basal membrane, then desmosomes, then tight junctions, then microvilli, then cilia, they are arranged. Okay, you can just have a look over this picture to get a feel. Understood? Now, basal lamina is connected to the epithelium with the help of finger-like digitations, which are known as our interdigitations. Okay. Then we have intercellular bridges as well, which are having intercellular matrix. Okay. So now coming to next, epithelial tissue classification. The classification of epithelial tissue is basically done on the basis of origin of epithelium from three primordial germ layers or germinal layers. Means, according to three primordial germinal layers, they basically give rise to different parts or different types of epithelium, and that is why they are classified in this way. Ectodermal, it gives epidermis, means stratified squamous epithelium. It is ectodermal in origin. Mesodermal means mesothelium, simple squamous epithelium. It is mesodermal in origin. Endodermal, endothelium, which is also simple squamous epithelium. So, ectodermal, it includes stratified squamous epithelium, and mesodermal means mesothelium, and endodermal means endothelium, which includes simple squamous epithelium. Classification of epithelial tissue on the basis of, this was the basis when on the basis of primordial germ layers. Okay, on the basis of primordial germ layers. Now we are going to classify them on the basis of numbers. Okay, when this classification is based on number of layers, whether the epithelial cells are arranged in a single layer or a multiple layer. So epithelial tissue, if they are arranged in a single layer, then simple epithelium. If they are arranged in multiple layers, then compound epithelium. If they are arranged in simple layers, then they may be known as pavement epithelium or squamous epithelium. Okay. Then columnar epithelium, then ciliated epithelium, and then pseudo-stratified epithelium. So epithelial tissue is basically categorized on the basis of number of layers in which the epithelial cells are arranged. First is the simple epithelium and the second is the compound epithelium. Simple epithelium is basically in which there is only one layer of cells which are arranged. It is of these four subsequent categories. One is the pavement epithelium or the squamous epithelium. Second is the columnar epithelium. And the third is the ciliated epithelium. And the fourth is pseudo-stratified epithelium. Okay? Pseudo-stratified epithelium. Understood? Now, the compound epithelium is further classified into stratified epithelium, Transitional epithelium. Compound means these are having multiple layers. The cells are arranged in a stack-like fashion, in one over the other. Okay, like for example, simple epithelium, the cells they are just having only one layer. If I make a plane like this, the only one layer of cells are present. Okay, these are arranged in a single layer, like pavement epithelium. If I draw a squamous epithelium like this, understood? Polygonal in shape, like this. Understood? And in case of compound epithelium, the layers are arranged. One layer, two layer, third layer, fourth layer. The cells are arranged. Understood? So this is a simple epithelium and the compound epithelium. Understood? The compound epithelium, they may be of stratified type or transitional type. Means stratified type, it is non-stretchable. It is non-stretchable. So I'll write here non-stretchable. And transitional epithelium, it is stretchable. It is stretchable. Okay. So the stratified epithelium, it is non-stretchable, while the transitional epithelium is stretchable. Stratified is further divided into stratified squamous and stratified columnar. Stratified is further divided into stratified squamous and stratified columnar. Okay. This stratified squamous is further divided into keratinized and non-keratinized. Okay. This stratified squamous is further divided into keratinized and non-keratinized. So this was the classification of epithelial tissue. Okay. This classification of epithelial tissue is basically based on the number of layers. Okay. Single layer or multiple layer. If single layer, then simple, multiple layer, then compound. And previous classification was based on the fate of primordial germinal layers. The ectodermal in origin means the epithelium which is of ectodermal in origin, which is basically originating or arising from the ectodermal cells. Epidermis, stratified squamous epithelium. Mesodermal is mesothelium, which is simple squamous epithelium, which is mesodermal in origin. Third, endodermal in origin is endothelium, which is simple columnar epithelium. Oh, so sorry. Please make a change. Please make a change. It is not squamous. It is not squamous. It is columnar. I'll write the whole simple columnar. Okay, so these are three different, classification of categories, and these were the categories in the different bases. Now understood? Stratified squamous epithelium, which is keratinized, means it is having a keratin layer. Okay, and stratified squamous epithelium, which is non-keratinized, means it is not having any keratin layer. Okay. Now coming to next, squamous epithelium. Squamous epithelium. The word squama basically comes from scales present on skin of fish. Okay, so these scales which are present on the fish skin, they are basically known as squama. Okay, or squamous. So basically, the word arises from that there only. They are unilayered. They are unilayered. Only single layer, and if they are single layer means it is simple squamous epithelium. Okay, simple squamous epithelium. Understood? The cells, they are flat or scale like in shape. The cells, they are flat, they are flattened or in a scale like of shape. A flattened or rounded nucleus is present. Nucleus is flattened or rounded and it is prominent. So a flattened, rounded and prominent nucleus is present. Cells are flat or scale-like. Cells appear rectangular in shape. Cells appear rectangular in shape. Okay. These cells, they are scale-like, as I have already mentioned, scale-like, and that is why their boundaries are irregular. Boundaries are irregular. Okay. Now, it is also known as pavement epithelium and tessellated epithelium. Okay. Tessellated epithelium. Tessellated. Tessellated epithelium. Okay. So please make a spelling rectification. Tessellated epithelium. Due to its wavy appearance means as it is so flexible that it gives a wavy appearance. Okay, and that is why it is known as what? Tessellated epithelium. This epithelium is also associated with filtration and diffusion. Okay, associated with filtration and diffusion. Understood? See, all the simple epitheliums, whether they are of different types, simple squamous, simple cuboidal, simple columnar, pseudostratified, their main function is as being as a lining of body cavities, ducts, and tubes. So I'll write in the previous slide. Just give me a minute. That all simple types of epithelium, okay, all simple types of epithelium, okay, I'll rub this part. All simple types of epithelium, all simple types, their main function is to act as a lining for body cavities, ducts, and tubes. Okay, so all simple type of epithelium, they act as a lining for all body cavities, duct, and tubes. Understood? And the compound epithelium, they mainly serve as a protective function as it does in our skin, in case of skin. So stratified, the main function is to protect. Sorry, protect. Understood? Now, this slide is already done. You can see in the picture, okay, this is a histopathological slide, okay, means under the microscopic section, you can see the boundaries of the cells, they are irregular, okay. The nucleus is very prominent and it is at the center. The nucleus is very prominent and it is at the center. You can see, okay, the cells are of different shapes, okay, pentagonal, hexagonal, and the nucleus is at the center and is prominent. Nucleus is very prominent. Understood? Now coming to next, where they are found. Okay, you can see the picture that this is the basement membrane. Basement membrane is a non-cellular, acellular layer, which is having the tessellated epithelium or the pavement epithelium. You can see the boundaries are irregular. Okay, nucleus is at the center, but they actually have very tight or compactly packing. They undergo very compact packing because of these cementing surfaces. Cementing surfaces. All these surfaces, they interdigitate in each other. Okay, that is why it gives a wavy-like appearance or a tessellated epithelium. They are present in boundless capsule, podocytes, descending limb, and thin part of ascending limb of loop of Henle. So please remember the locations you have to remember. Bowman's capsule, they are present in Bowman's capsule, podocytes, okay. Secondly, descending limb and thin part of the ascending limb of loop of Henle, okay. Retitestis, eye lens of retitestis, in retitestis they are present, eye lens, alveoli of lungs, pneumocytes, okay, or air sacs. Alveoli of lungs, pneumocytes or air sacs, they are present squamous epithelium. They are also present in small bronchioles. Okay. Now, very important. Bowman's capsule or podocytes, they basically help to, filter the blood for further, in the process of formation of urine. The blood needs to be filtered from all the substances which are less needed or not needed. Okay. So body is going to reabsorb all the substances which are required by the body, while the substances which are not healthy for body, they are going to be eliminated. So you have to remember the exact location that this simple squamous epithelium is present on those respective sites which need high amount of flexibility, which need high amount of filtration barrier, okay, or high amount of diffusion barrier, okay. So all these places, they actually require high filtration or diffusion. The main examples are this podocyte, descending limb, okay, and the thin part of the ascending limb, then retitestis, alveoli of the lungs, and yeah. Now mesothelium, mesodermal in origin. Covering of the coelom is called mesothelium. The coelom is covered by a part which is known as what? Mesothelium, and it is known as what? Tessellated epithelium. Okay. So mesothelium basically it covers the coelom and it is called tessellated epithelium. Okay, understood? Yeah. Visceral and parietal peritoneum. Gut cavities, the cavities of the alimentary canal, they are known as what? Peritoneal cavity. The visceral and parietal peritoneum, they basically covers the linings of organs. The visceral and parietal pleura, visceral and parietal pericardium. So you can see almost all the organs, they are basically covered by these layers. Parietal means outer and visceral means inner. Outer and visceral means inner. Understood? Endothelium, it is the inner lining of the blood vessel or lymph vessels. Endothelium, it is the inner lining of the blood vessels or the lymph vessels. It is also tessellated. Also tessellated. Means endothelium, mesothelium, they all are tessellated. Okay. And epithelium also, it is also tessellated. Understood? So endothelium, the inner lining of blood vessels and lymph vessels, it is also tessellated type of epithelium. Understood? Now, inner lining of heart, inner lining of heart, it is also tessellated epithelium. So just remember the examples: Bowman's capsule, podocytes, then alveoli, in air sacs, okay, or the pneumocytes, descending limb and the thin ascending limb of loop of Henle, retitestis, islands, mesothelium, okay, it is also a tessellated epithelium. Then visceral and parietal peritoneum, visceral parietal pleura, visceral parietal pericardium, all are tessellated epithelium. Endothelium, tessellated epithelium, okay, and inner lining of heart bone. They all are made up of simple squamous epithelium. Now, simple cuboidal epithelium. The cells are cube-like in shape. Cells are cube-like in shape. Basement membrane is present. Okay, so in case of simple cuboidal epithelium, you can see that basement membrane is present, and most importantly, cells are cube-like in shape. A rounded nucleus is present in the center of the cell. A rounded nucleus is present in the center of the cell. Cells are of the same in length and width. They are cube-like, cubical. So they appear square shaped in vertical section. When we look in the vertical section, they look square shaped. But when we look in the other sections, in the other projections, they look like a cube. Okay. This epithelium helps in absorption, secretion, and excretion. The main function of this type of epithelium, the cuboidal epithelium, is absorption, secretion, and excretion. Okay. This epithelium is also called the germinal epithelium. Okay, it is also known as the germinal epithelium. Okay, so this epithelium helps in the absorption, secretion, and excretion. It also forms gametes in gonads. That is why it is known as what? Germinal epithelium. Germinal epithelium. Mostly cuboidal cells are found in glands. Mostly cuboidal cells are found in glands. Understood? Maximum of the glands which are present in our body, they are basically having these cuboidal cells because they are rich in absorption, secretion, and excretion of the substances. Vesici- vesicles vesicles of the thyroid gland, the vesicles of the thyroid gland, pancreatic duct, secretory unit of sweat glands, secretory duct of salivary glands. Secretory unit of salivary glands is composed of stratified cuboidal epithelium. So please remember, the secretory duct of salivary glands and secretory unit of salivary glands, they are two different things. The secretory duct of salivary glands, with the duct of salivary glands, it is having a covering of simple cuboidal epithelium. While the secretory unit of salivary gland, it is composed of stratified cuboidal epithelium. It is composed of stratified cuboidal epithelium. So, the vesicles of thyroid gland, pancreatic duct, secretory unit of sweat glands, secretory unit duct of salivary glands, not the secretory unit of the salivary glands, it is secretory duct of salivary glands. Okay. Iris, choroid, ciliary body of eye. Okay, so iris, choroid, ciliary body of eye, thick part of ascending limb of loop of Henle. Okay, thin part of ascending limb of loop of Henle was basically having the simple squamous epithelium. Thick part of ascending limb of loop of Henle was having simple cuboidal epithelium along with DCT, distal convoluted tubule. Okay, so iris, choroid, ciliary body of eye, thick part of ascending limb of loop of Henle, and distal convoluted tubule, they are basically covered with simple cuboidal epithelium. In gonads, this epithelium is called germinal epithelium, testis and ovaries. Their cuboidal cells divide to form egg and sperm. Okay, the cuboidal cells, they divide to form egg and sperm. That is why it is known as what? Germinal epithelium. It is found in the peripheral region of the ovary. It is found in the peripheral region of the ovary and in the wall of seminiferous tubules in the testis. Okay, so these are some common locations of simple cuboidal epithelium. Okay, so the simple cuboidal epithelium, it is present in vesicles of thyroid gland, ascites of pancreas, secretory unit of sweat glands, secretory duct of salivary glands, then iris, choroid, ciliary body of eye. Yeah, means at the ascending limb of thick part of the ascending limb of loop of Henle and DCT. Along with the germinal epithelium, it makes or that helps in the production of gametes. Okay, understood? It is also found in the peripheral region of ovary and in the wall of seminiferous tubules. Okay, so this is the example of simple cuboidal epithelium. Understood? Now coming to next, modifications. The simple cuboidal epithelium, it undergoes modifications to form brush border cuboidal epithelium and the ciliated cuboidal epithelium. The brush border cuboidal epithelium, where microvilli are present on the free surface of the cuboidal cells. PCT of nephron, very important. The proximal convoluted tubule of nephron is basically having a modification of the simple cuboidal epithelium, in which there is the presence of what? Brush border epithelium. And brush border epithelium is a modification of simple cuboidal epithelium. It is a modification of simple cuboidal epithelium. Ciliated cuboidal epithelium. Ciliated cuboidal epithelium, when cilia present on free end of cuboidal cells, then collecting duct or tubule. So brush border epithelium and ciliated. Brush border epithelium is present in the PCT. Okay, free surface of the cuboidal cells, they are having microvilli. PCT of nephron. And ciliated cuboidal epithelium, when cilia present on the free end of cuboidal cells, then the collecting duct. Examples are collecting duct and tubule. Okay. So what I am just telling, please just listen. Descending limb of loop of Henle, This is the nephron. Okay. So the proximal convoluted tubule, the descending limb. Okay. This is the thick segment of the loop of Henle, which is the descending limb. Descending limb. This is the ascending limb. Okay. So just remember, you can see the pictures. The proximal convoluted tubule, it is made up of the ciliated, so not ciliated, brush border epithelium, which is having microvilli, finger-like projections, which are non-motile. Okay. Ascending thick segment of loop of Henle. This is the ascending thick segment of loop of Henle, which you can see it is having simple squamous epithelium. Simple squamous epithelium. Ascending thin segment loop of Henle. Okay, this is ascending thin segment of loop of Henle. Collecting duct, ciliated cuboidal epithelium. Collecting duct, sorry, distal convoluted tubule. The collecting duct, it is also having the ciliated cuboidal epithelium. Okay, ciliated cuboidal epithelium in case of distal convoluted tubule, DCT, and the collecting duct part. Okay, so you can see the pictures now. I'll just move on the previous slides to avoid the confusion. Just a minute, just a minute. Yeah, descending limb and thin part of ascending limb of loop of Henle, they are made of simple squamous epithelium. Okay, descending limb, this one, and the thin part of the ascending limb, they are made up of squamous epithelium. Okay, thick part of the ascending limb is made up of cuboid, the simple columnar epithelium. So here I'll mention the change. This is made up of squamous epithelium. This is also made up of squamous epithelium. Thick part is made up of cuboidal epithelium. Cuboidal epithelium. Okay. Collecting duct, especially the terminal part of the collecting duct, I'll write here, the terminal part of the collecting duct and DCT. They are basically having the ciliated cuboidal epithelium. So please remember, DCT, ciliated cuboidal epithelium. Understood? TCT is having brush border epithelium, DCT is having ciliated cuboidal epithelium. Squamous epithelium is present in those parts, the descending loop and the thin part of the ascending loop. Okay, now it's clear. Now these are microvilli, okay, which are basically having the blood vessels in the folds of intestines for the absorption of food. Okay. Now coming to next, simple columnar epithelium. Simple columnar epithelium is basically cells are pillar-like in shape, tall and slender. Basement membrane is present. Cells are pillar or column-like in shape, tall and cylinder. Nucleus is elongated and present at the base of the cell. Nucleus is elongated and is present at the base of the cell. Main functions are absorption and secretion. Main functions are absorption and secretion. Examples are bile duct of liver, bile duct of liver. Examples are bile duct of liver. Okay, it is misprinted 2 times. Okay. Modifications. Brush border columnar epithelium. Brush border columnar epithelium is basically when microvilli are present. Gallbladder. Example is gallbladder. So microvilli are present at the free end of the epithelium. Gallbladder is the example. Glandular columnar epithelium, in which unicellular mucus secreting goblet cells are present. Unicellular mucus secreting goblet cells are also present in between the columnar cells. So in between the columnar cells, there is a presence of mucus secreting goblet cells, right? So example, stomach, colon, rectum. So there are 2 modifications you can see. Furthermore, we are going to talk about. First is brush border, in which the microvilli is present on the free end of the epithelium. Example, gallbladder. Example is gallbladder. Second is glandular columnar epithelium, in which unicellular mucus secreting goblet cells are present in the columnar cells, okay, like stomach, colon and rectum. Understood? Then the glandular or brush border columnar epithelium, okay. glandular or brush border columnar epithelium. Wait. Yeah. Glandular or brush border columnar epithelium in which microvilli are present on the free end of columnar cells, and in between these cells, goblet cells are present. In between these cells, goblet cells are present. So this is known as what? Glandular brush border columnar epithelium. It is present in duodenum and ileum. Duodenum and ileum. Okay, duodenum and ileum. Understood? The duodenum, ileum, cecum, they are having glandular brush border columnar epithelium. Stomach, colon, rectum, and uterus, they are having glandular columnar epithelium. And brush border epithelium, gallbladder. Okay. Now, ciliated columnar epithelium, when cilia are present, example, fallopian tube and ependymal epithelium of brain ventricles. Fallopian tube and ependymal epithelium of brain ventricles. Understood? Tympanic cavity of middle ear and auditory tube. Tympanic cavity of middle ear and auditory tube. Understood? So, I'll revise it for you again. Just give me a minute. Yeah, modifications. The brush border columnar epithelium is present in gallbladder, glandular columnar epithelium, stomach, colon, and rectum. Then, brush border glandular columnar epithelium, duodenum, ileum, and cecum, ciliated columnar epithelium in fallopian tube, ependymal epithelium, tympanic cavity, and auditory tube. Understood? This is the ciliated columnar epithelium with the goblet cell. You can see the mucus producing cells, these are goblet cells. Goblet cells. Okay? And these are the simple columnar epithelium, they are also having the goblet cells. Okay, these are known as what? Goblet cells, which are having the mucus producing phenomena. Okay, goblet cells. Understood? Now coming to next, you can see the picture very well. Pseudo-stratified ciliated columnar epithelium. Okay, so before starting the pseudo-stratified ciliated columnar epithelium, there is one more modification of the columnar epithelium was stereociliated columnar epithelium. Stereociliated vasa differentia and epididymis, only two junctions in the body which are having the stereocilia, false cilia. Stereocilia present on the free end of the columnar cells. Okay, epididymis and vasa differentia. Now, pseudo-stratified ciliated columnar epithelium, it appears bilayer as two types of cells are present. Okay, very important. The pseudo-stratified ciliated columnar epithelium, it appears bilayered. Well, it looks like that it is having two layers. One layer is made up of long cells which are having oval nuclei, and one layer is made up of short cells which are having round nuclei. Okay. So the cells which are having oval nuclei are the long cells, and the cells which are having round nuclei are short cells. But the fact is, both the cells don't touch the outermost part, periphery. In only long cells, they are able to touch the outermost part of the periphery, the short cells not. Okay. But all the cells are present on a single basement membrane. Long cells and short cells, they are present on a single basement membrane. So it is unilayered. That is why it is unilayered. So pseudo-stratified epithelium, it looks like that it is having two layers. The long cells which are having oval nuclei and the short cells which are having the round nuclei. But all the cells, they are present on a single basement membrane. So it looks like that it is unilayered. Okay. It is of two types, pseudo-stratified columnar epithelium. Pseudo-stratified columnar epithelium, example, parotid salivary gland, middle part of male urethra. Okay, so parotid salivary gland and middle part of male urethra, they are having pseudo-stratified columnar epithelium. Pseudo-stratified ciliated columnar epithelium, pseudo-stratified ciliated columnar epithelium. Okay, it is also present. In this epithelium, cilia are present at the free end. Okay, so one is non-ciliated, one is ciliated. The non-ciliated is present in parotid salivary gland and middle part of male urethra, and ciliated columnar epithelium, pseudo-stratified, is present in the trachea, bronchi, respiratory epithelium of nasal chambers. Okay, So the pseudostratified epithelium, columnar epithelium, is basically having two categories: the ciliated one and the non-ciliated one. Non-ciliated one, they are present in the parotid salivary gland. And the ciliated one, they are present in the trachea, bronchi, respiratory epithelium, because they are having the goblet cells, and the goblet cells, they also produce mucus. And that is why these cells, they secrete mucus to eliminate any dust particle or any unwanted foreign material, which actually is going to or try to enter the respiratory tract. That is why mucus is bad for us, mucus is good for us, and ultimately mucus is protective for us. Because of this mucus only, the dust particles, they cannot enter the deep spaces of our body. They are not able to enter the respiratory, epitheliums, and that is why this mucus acts as a protective layer. So these are some common different categories of simple epithelium I discussed with you. Okay. After simple epithelium, I discussed the pseudostratified ciliated columnar epithelium. Okay. Pseudostratified ciliated columnar epithelium. Understood? So I'll just revise for you that simple columnar epithelium, it is basically of brushwater epithelium modification, glandular columnar, glandular brushwater columnar, then the ciliated columnar, and then stereociliated columnar. These were the five modifications of simple columnar epithelium. Okay? Then we have learned about the cuboidal epithelium. Cuboidal epithelium is further, modified into brushwater cuboidal and ciliated cuboidal. Okay. Then squamous epithelium. Squamous, squamous epithelium is actually not having any modifications actually noticed, but the sites where they are present, it is very important. Like Bowman's capsule, podocyte, alveoli of lungs, then the descending limb and the thin part of the ascending limb, all these parts, please remember mesothelium, endothelium, they all are having the squamous epithelium. And it is also known as the tessellated epithelium. Understood? So please remember all these terms. Now coming to next. Yeah. Spatial types of epithelium. The neurosensory epithelium or the neurosensory cells, which are present in between pillar-shaped supporting cells, modified sensory cells are present. Okay. The neurosensory epithelium, it is basically a pillar-shaped supporting cells, or we can say they are modified sensory cells. On the free end, sensory hair is present, and base of these cells is attached with the sensory nerve. Base of this cell is attached with the sensory nerve. Okay. So neurosensory epithelium, it is epithelium. Epithelium. These neurosensory epithelium, they are special types of epithelium, which are basically in a pillar-shaped supporting cells, which are having the free ends, which carry the sensory hair. Okay, and the base of these cells are attached to the sensory nerves. Like for example, gustatory epithelium, taste, cover taste buds of the tongue and receive taste sensation. You can see the picture, the sensory hair. You can see they are pillar-like. Okay, these are the columnar cells, and these are the pillar-like. Okay, then they are having the nerve fibers attached to them. These are neurosensory cells. Olfactory epithelium, Nerdian membrane, receives smell sensation. Statoacoustic lining of internal ear. It is also a neurosensory epithelium. Statoacoustic. Stato means balance, and acoustic means hearing. The internal ear, the ear, basically helps as a balancing organ as well as the hearing organ. Okay. During any trauma or accident, when a person hits, very drastically, very impactfully to some other object or some other surface, so what will happen? The equilibrium gets disturbed. The body equilibrium gets disturbed, and he got or she got fainted. That is because of the disbalance in the statoacoustic sensations. In retina of the eye, receive optic sensation, neurosensory epithelium. Myoepithelium, around mammary and sweat glands. It is also a type of special type of epithelium, myoepithelium. Understood? Then, pigmented epithelium, it is a modification or the special type of cuboidal epithelium in retina of the eye. So you can see the neurosensory types of epithelium, they are having, modified, columnar cells. And the myoepithelium, it is present around the mammary and the sweat glands. It is also a special type of epithelium. Then the pigmented epithelium means the cuboidal epithelium. It is also a special type of epithelium which is present in the retina of eye. Okay. So we will be starting the compound epithelium in the next turn. I hope everything is understood. Okay. Doubts are always welcome. Please go through the previous slides to actually have a connect to the upcoming video lectures. Till then, bye bye. Take care. Stay safe. Stay at home.